Thank you. It's an honor to be here to talk about genome engineering. And also, I'm very pleased to really have my first acquaintance with the Royal Canadian Institute for the Advancement of Science. I didn't know very much about this organization before, but what I've heard in the last little while has really been impressive. And I think that we're at a point now where science advocacy is really going to be effective. Okay, so disclaimer here, as mentioned, as well as being a professor at Waterloo, I'm also co-founder of this company, Metagenome Bio. The work that I do is not all genomics first and foremost, but as microbiologists, almost everything that we do is influenced by genomics because all of the organisms that we work with have genome sequences available. And we also work with organisms in the environment that we don't culture, and we detect them by DNA sequencing, and the genomic information helps us here. So what this does, because I won't have an opportunity during the talk, is just tell you a little bit about what I do in my research lab. And the talk is going to be a mix of what I do in my lab and then also some of the hot topics as far as genome engineering go. So I first got started working with this fascinating microbe. Does anybody know what this is? This is bacteria that are in root nodules. So root nodules are where symbiotic nitrogen fixation happens. And it's where the nitrogen in the air is converted into ammonia. And then that feeds the plant, and so almost all legumes are able to do this. These are alfalfa root nodules. So we study this, we learn about the genetic interaction between those organisms, and hopefully in the future, maybe we'll be able to engineer them to do this work better, or some people are working on trying to get rice and corn and other types of non-legume organisms to form these partnerships and also fix nitrogen. We also do, so here's a soybean field. We got interested in doing work on microbial communities. All of that work is genomic in nature. So basically you can take a fingerprint of the soil by looking at the DNA sequence and you can tell what types of organisms are present. This is something that is not very pleasant, but you've probably all seen this before. These are plastics. Our life is inundated with plastics. They're very important for us. But wouldn't it be great if they were biodegradable? It turns out that there are some bacteria, such as the ones that make these root nodules, and a lot of other ones, that make these deposits within the nodule that can be used, that can be extracted and used as biodegradable plastic. So we're trying to figure out better ways to make these plastics and make plastics with more beneficial properties. And then this is meant to signify some of the genome engineering that we do. Not all of our work is agricultural. We also do some work with human digestive system. So this is meant to be fecal matter here that we extract or that comes out naturally. You can actually get a lot of information about what's inside your gut from sequencing those organisms. So that's sort of the different types of work that I do in my lab. And here's another disclaimer. It's really important for academic scientists to say where they get their funding from because there's a lot of sense that whatever they say might actually be colored by their funding. So of course we do get some commercial funding and have in the past, and really you have to in order to run a research program. So here are some of the companies that have funded us, as well as the funding organizations, the public funding organizations, but for the most part, these are small amounts of funding. So Polyfirm is a small bioplastics company, Novozymes is a big enzyme company, MedRelief is a startup medical marijuana company, and that's about it. You can ask me about the medical marijuana and what we're doing with it later if you'd like, after the talk. Okay, so this is where we come from as scientists. This made a really huge impact on me. This shows an estimate of how much food we're going to have to produce in the next 50 years in order to feed the growing population. We are going to have to produce more food in the next 50 years than has been produced in all of human history. So let that sink in a little bit. That's really astounding. How are we going to do that? I'm confident that we will be able to do it, but the question is, how are we going to do it? I think that we're going to need all of the tools at our disposal. And I think that some of those tools will include things like genome engineering. So let's look at what genome engineering is and how it might actually be harnessed to address this problem. A little bit of context for what's been happening in agriculture in the last number of years. Here we have cereal yields from 1960 to the present, and this is for developed countries, for basically North America, Europe. You can see quite an advance there. Asia and Latin America, those parts of the world are all doing quite well. But look at the cereal yield in Africa. Basically, it's not going anywhere. It's improving a little bit, but not really that much. There's a huge potential here. Are we going to be able to do that? That's where there's a lot of mouths to feed. It would be great if they could produce enough food to sustain themselves. That's a major challenge. Here's another statistic here. Worldwide employment in agriculture. Here we have the developed world. We have where are we? Oh, sorry, I'm going the wrong way. We have the developed world here. Not very many people in agriculture. Here we have Latin America and the Caribbean. A few more, as this is percentage here. But look what happens when you get up to sub-Saharan Africa. The majority of the people are actually working in the agricultural system. But they're working and the productivity is not that great. It takes a lot of people to produce not that much food. If you have a chance to take a look at this book here by Robert Parlberg called Starve for Science, I think it's worthwhile to read this extract. So let's go through what he says. And I was just reading this last night, partly in preparation for this talk, but also partly because it's an incredibly important area. Okay, so what he says is the economic advantage of a highly productive, technologically advanced farming system is its capacity to bring down the cost of producing food while simultaneously releasing workers for employment outside of agriculture in the industrial and service sectors of the economy. The post-industrial affluence enjoyed today by urban and suburban dwellers in both America and Europe rests on those regions' much earlier adoption of science-based farming. So notice it says here science-based farming. That's what's actually resulted in that productivity, bringing science into agriculture. There's a lot of innovation in agriculture that people aren't aware of. The other thing here is in the United States, and the same for Canada, the percentage of citizens employed in agriculture fell from more than 50% in 1870 to only 2.8% by 1990. What we are able to do in modern civilization, our educational systems, our businesses, a lot of that is because we've been able to become more productive on the farm. But there's some parts of the world that are left out by that, and let's give them a chance to catch up. Okay, another important thing that is often neglected is this. This shows, remember those root nodules that I talked to you about? They fix nitrogen. They convert the nitrogen in the air, so the most abundant gas in the air is N2 gas. They convert that to ammonia, and then that can be taken up by a biological system. So there are no biological systems except bacteria that are able to do this. Some of them work in these root nodules with these with these plants, but some of them also work in other ecosystems. So this is the baseline of biological nitrogen fixation. Over 100 years ago, there was the Haber-Bosch process, which basically takes hydrogen from methane, from natural gas, and under high pressure and temperature, produces, from the N2 gas, produces ammonia. Okay? And that's where most of our fertilizer comes from. So it's a high energy process, requires a lot of natural gas. But look what happens to the amount of nitrogen that's present in our environment. Do we think that there's going to be no environmental effect of having all of that extra human-made nitrogen in the environment? Well, of course there are problems with that. But that has really been the basis of the green revolution. It's the basis of the productivity of our crop plants. That and also the better genetics in the seeds. Well, this has had some unintended consequences. How many of you heard of the dead zones? So we know that in some areas here, this is near the Mississippi River Delta. The red areas are areas that are more oxygen deficient. And the reason that they're oxygen deficient is because, of course, all of that fertilizer that isn't used by the plants goes into the rivers. It comes down to where it's output here. And what happens there is that you have these blooms of bacteria, and they use up all the oxygen. So they're not dead because there's no life there. There's actually a lot of life. There's a lot of microbes, but they use up the oxygen so that things like fish are not able to survive. Well, that happens because we've been applying this fertilizer, and the plants aren't as efficient at taking it up as they could. If we were growing only soybean and not corn, then we wouldn't really have that problem because you don't have to fertilize soybean with nitrogen fertilizer because it's making its own. So wouldn't it be great if we could get corn to produce its own fertilizer? Well, if you look around the world, look at where all of these dead zones are concentrated. And notice that there's not very many around Africa. It's because they don't use very much fertilizer, because it's actually quite expensive. And that's one of the reasons why their productivity isn't great. I'm going to move from agriculture, but I'm going to come back. I just wanted to spend a couple of minutes talking about plastics. So, 10% of our fossil fuels are used to make plastics, but I don't think that's really the big problem. The big problem is that about 20% of our landfill sites is plastics, and a lot of that actually makes it out into marine environments, into freshwater environments. We have this problem in the Great Lakes as well. The plastic doesn't decompose, and a lot of it is hidden just below the surface, so you're not aware that it's there unless you actually look. So can we take advantage of genome engineering in order to solve this problem? I'm not going to give you too many details of that. Actually, I'll come back to that later, now that I see this slide. So what is the goal? What is our overall goal? Is to produce safe and nutritious food and materials for the growing world population while minimizing increases in land use and reducing environmental degradation. So that's really what drives me as a scientist. I'm interested in fundamental research, but I'm also interested in how that can be applied to solving these big problems. And I think a lot of scientists are driven by that. Okay, so first, this is supposed to be a talk about genomes and genome engineering. So just so that we're all on the same page, what is a genome? So a genome is the complete set of genetic material within an organism. These vary quite a bit in size. If you look at the wheat genome is huge. It's one of the largest genomes, and that's because wheat has been bred for 10,000 years or so, and it actually is an amalgam of 3 separate genomes that have come into one. Genome engineering is really the precise manipulation of the genome. So can we change the genome based on our knowledge of genomics, so based on our knowledge of what those genes are doing, in order to produce organisms that have beneficial properties? And this is an important part of something that we call synthetic biology. Synthetic biology is really building organisms that can carry out processes that you want them to do. Okay? That's sort of a general description of what synthetic biology is. Okay, we've been doing genome engineering for a long time. This is from work that I did as a graduate student over, well, let's say a quarter century ago. And we were able to do things like make deletions in the chromosome using techniques that were classical genetic techniques. They didn't involve any recombinant DNA or anything like that. They involved things like transposons that are these pieces of DNA that are able to move from one part of the genome to the other and from one genome to the other. And we made use of things like homologous recombination in order to delete out regions. So you can see here, this region here, through this genetic cross that has the segments B, C, and D, are absent in this deletion. And we did a few other crazy things, like took two adjacent deletions and combined them so that they were bigger, and reduced the deletion size, all sorts of fun that we had, until we were able to take this bacterial chromosome, and each of these indicates a deletion of the chromosome. And people have continued to work on this, and they've actually been able to build an organism that has a highly reduced genome, but it still functions. This is one of the 3 chromosomes in the bacteria, and you can see you can get rid of almost every part of it. There is another chromosome you could get rid of completely, and then the largest chromosome, you could get rid of part of it, so that eventually you have a strain that is deleted of more than half of its genome. Okay, so we're talking about bacteria here. Bacteria are one of the branches of life. We also have archaea, which are also microbes, and then we have eukarya. Most of the eukarya are microbes. Only a few are things like animals and plants and insects and those types of things. But they're all sort of clustered here at the top. There's a huge diversity of life out there for which we don't really understand very much. And often people aren't aware of them, they don't talk about them very much. One of the things that people aren't aware of is that it's not a simple tree, that we actually, through evolutionary processes, there have been transfers of DNA, natural transfers of DNA encoding genes between these different branches of the tree. So we actually have genetic chimeras. All of you are genetic chimeras. You're made up of DNA, your genome is made up of DNA that comes from many different sources. Your mitochondria come from a certain type of bacteria that's actually related to those root nodule bacteria. You don't have chloroplasts. I was going to say that you have chloroplasts, but you're not plants. But the chloroplasts of plants come from another type of bacteria, which are related to the blue-green algae. Okay, so the reason I was showing you that is because I wanted to tell you about the huge amount of genetic information that we can actually get from microbes. Most of the microbes we don't know, and we can't cultivate them as microbiologists. So the only way that we can study them is we can go out into the environment. So you go out to one of these environments, you extract some soil, you take the DNA out of the soil. There will be millions of bacteria and other microbes present in the soil. That DNA represents the genetic potential within those genomes. So here are some of the places where we have isolated, where we've taken soil and we've isolated the DNA. And you can see that Canada has a huge diversity of soils, and those soils are represented by a huge diversity of genetics, a huge diversity of different types of organisms. So this is a fingerprint that we can do showing, by DNA sequencing, showing the different taxonomic classes of organisms that are present within those different soils. So why am I telling you this? I'm telling you this because I want to tell you about how we can actually access that information and use it in order to produce something that's interesting, that's useful, like a bioplastic. Okay, so we can make these libraries, and Cheng here in my lab is a master at making these libraries. This wheat field soil is our best library. It has 9 million clones. It's a large number of clones. And we use this vector here. Basically, this is just a plasmid, a piece of DNA that we can take the DNA that we extract from those soils, take the fragments and clone them into there. So each clone has a piece of DNA from a different part of a different genome. Okay? So basically what we've done is we've taken that DNA and we now have a copy of it in our freezer. So we can then see if we can isolate genes for bioplastics. Why would we want to do that? Well, one of the reasons we would want to do that is because different bioplastics have different structures. And the structure is determined by the carbon compounds that you give the bacteria to eat, but they're also determined by the enzymes that are encoded by the genes that actually make the polymer. Okay? So these are polyesters. And you can see that there's some diversity indicated by this N here and by this R group. So that basically this R group can be a number of different things. And so that type of diversity is what we're interested in. And I won't go through this biochemical pathway, but I will tell you here, this is the important thing, is that this is the strain and the type of plastic that's normally made. When you take some of the DNA from the environment and you put it in the organism, you can actually make plastics that are different, and they have different physical properties. And when you look at the DNA sequence of the genes that encode those enzymes, they're different than what you might expect. So that's an example of how you can actually mine the genetics of the environment. Okay, so we're talking about genome engineering. It's important that we put this into a little bit of historical context. So this really got started in the 1940s. It was started, a lot of the work was done by a Canadian from Halifax named Oswald Avery, who was working at the Rockefeller Institute in New York City. And he showed that DNA actually carried the heritable information, okay? That DNA carried genes. There's a lot of opposition at the time. A lot of people thought that DNA was too simple, but through some very simple experiments, but using stringent criteria, he was able to show that it was actually DNA that was doing this. Okay, so we go from there. There's a lot of molecular biology that was started up at this time. DNA sequencing was invented. Site-specific mutagenesis and the ability to synthesize genes was invented at this time. There's another Canadian who was involved in site-specific mutagenesis. Does anybody know who that is? Nobel Prize? Michael Smith, who worked at UBC at the time. And he was actually did some work with Gobind Khurana, who actually invented gene synthesis. There is a bacterial genome sequence, which happened here. iGEM, which I'll tell you about. This is an undergraduate student competition where they do synthetic biology. And I'm going to talk to you a little bit through the rest of this talk about genome transplantation, genome synthesis, and CRISPRs. Okay, so it's still a little bit of historical work here. A lot of people talk about one of the first successes of recombinant DNA as being the production of insulin, recombinant insulin. What a lot of people don't understand is that in this example, the DNA was not just cloned from human cells. They actually had the protein sequence, and they synthesized the DNA that would produce the protein. So it was a synthesis event where they chemically synthesized using 29 short pieces of DNA, averaging 14 nucleotides in length. That's a total of 181 base pairs of sequence. It's not really that much, but it was enough for the two chains of human insulin. That took 4 workers 3 months. Today, that would be less than a $100 order, and it would probably be shipped in a couple of days. So we've advanced quite a bit from this time, and one of the things that's happened is that it's now possible for almost any research lab to work with synthetic DNA, and it's changed the way that scientists have worked. So this is called the Carlson Curve, which is data that's been compiled by Rob Carlson, and this shows the cost of sequencing. Sequencing is basically free at this point. You can generate DNA sequence data without even thinking about the cost. It's the cost of analyzing that sequence data that's important. But this is something that's maybe perhaps a little bit more interesting. This is the cost of gene synthesis. Pretty soon, gene synthesis is going to be something that you don't actually think of the cost. So what are the implications of this? If you think about it, what this means is that you can have an idea of the sequence of a gene according to the properties that you understand through the biology that you are researching, and you can have that DNA synthesized even though it might not exist in nature. Okay, so what are the implications? What can you do with that? There's a lot that you can do as far as research goes. There's also a lot that you can do as far as industry. And there's a number of gene synthesis companies. Now, for the average gene in a bacteria to have it synthesized, it costs about $200. And this was taken advantage of by Craig Venter and his team. So 2010, so about 5 years ago, this made a big splash. This is from New York Times. Scientists say they created a synthetic cell. And this is from the actual article that was published, Creation of a Bacterial Cell Controlled by Chemically Synthesized Genome. Many of you are probably aware of this work. Basically, they were working in an organism called Mycoplasma mycoides. The reason they used this organism is because it has a pretty small genome. And basically what they were able to do is make small pieces of DNA that was a sequence that was very close to the naturally occurring wild-type Mycoplasma, sorry, that should be Mycobacterium mycoides. To make fragments that would overlap. They then stitched them together using different recombination techniques, but the real trick came when they were able to put these into yeast and use the recombination system within yeast in order to stitch together the parts until they had a full-size genome. And then extract that DNA from the yeast and transform it into a recipient cell. And here they're putting it into a Mycoplasma cell that's of a different species because they have different types of selection that they can use, antibiotic resistance selection. They're able to retain only the synthetic DNA, which then displaces the DNA that was already present. And then they would determine the sequence of that, and they would show that it actually was the synthetic organism. So you can think that now, with that capability, you should be able to make almost any organism that you want according to specifications. And they've continued doing this by improving the ability to take different types of bacterial DNA and clone it into yeast. And there are some companies, including this new one here called Designer Microbes, which is based in London, Ontario, which have arisen based on this. And there's actually a large number of companies, mostly in the United States, which are producing novel organisms to specification to carry out different processes. And here's an example. Amyris technology makes a lot of different types of compounds using synthetic organisms. So you can think of different food ingredients, fragrances, flavors, pharmaceuticals, lubricants, polymers, and fuels. And basically what they're doing is they're converting sugars into these different compounds through fermentation processes. Okay, so let's get to plants. Okay, so a lot of people are interested in plants and they're interested in what they're eating. We've been breeding crops for 10,000 years. Here's a great example. Wild mustard gave us cauliflower, gave us broccoli, cabbage, kale, kohlrabi. These things, we sort of know that they're related, but do we know that is it evident that they all came from wild mustard? Looking at them, it might not be so apparent, but we've had a lot of time in order to develop these types of plants. Plants do a lot of really interesting things, and bacteria that interact with plants also do a lot of interesting things. The reason why I am a microbiologist is because I fell in love with this interaction here. This is a tumor that forms on a plant. In this case, it's a tobacco cell. I think that's a tobacco, oh no, it's not a tobacco cell. But often tobacco cells are used, and it's sort of ironic that people study an organism that gives tobacco cancer. But a lot of the early work was done actually on tobacco. Here's out in the wild, this is my graduate student Kathy Lamb, who discovered some tumors near Halifax when she was away at a conference. Those could be agrobacterium tumors, which this is, or they could be Pseudomonas tumors, or they could be nematode tumors. There's a number of tumors that form. The agrobacterium are the ones that we're interested in, because what the agrobacterium does is it actually transfers a piece of its DNA into the genome of the plant cell. And after it does that, the genes are expressed, the genes from the bacteria are expressed, and they actually cause the production of different types of hormones. And these are plant hormones, and that causes the cells to grow and divide, and that's why you have a tumor. But the agrobacterium also produces, it gets the cells to produce a weird type of amino acid called an opine that only that bacteria can eat. So basically, agrobacterium is genetically engineering the plant to make a home for it and also to make food for it. Okay, so it's a fascinating interaction. And the most fascinating part of it is the fact that the DNA is transferred. And this is related actually to what I was showing in one of the earlier slides, where you can see that there's lateral gene transfer between the different types, between the different branches on the tree of life. Okay, some of it happened this way. There was this paper that came out recently which showed that the genome of cultivated sweet potato, all cultivated sweet potato actually has agrobacterium sequences in it. So it's naturally being engineered by agrobacterium. We've taken advantage of this. Actually, this is, I went to work with Gene Nestor when I was doing a postdoc at University of Washington in Seattle. And here, you know, this was a big splash just in the last year or so because it's in a food crop. But many, many years ago, this was in 1986, Gene Nestor and his team showed that the same thing was happening in Nicotiana, which is tobacco. Okay? It's not a food crop, so people didn't really care about it too much. So this is actually the basis of plant genetic engineering, the making of transgenic plants, is the ability to co-opt the system, so this plasmid here, and take out those genes that make the plants form a tumor, take out the genes that make it produce the food for the bacteria, and substitute other genes. So some of the genes that they've substituted are genes for Bacillus thuringiensis, or BT, anti-insect compound, which is taken up by certain insects, and it basically blows up their guts so that they no longer survive. Or for different types of herbicide tolerance. Those are the main traits. And this is really an old technology. These traits were first introduced a couple of decades ago. And that's usually what people think of as transgenic plants. Okay? But it's an old technology. Agrobacterium isn't the only way you can do it. You can also use a gene gun. This has actually been something that has been a big part of the productivity increases in North American crops. The ability to easily get rid of weeds that compete for resources and not having to spray with insecticide. And it's fairly simple to do. Basically, you can punch a hole out of the leaf. You incubate it with the agrobacterium cells that have the proper constructs within them. Those can regenerate because you can put them into different types of hormone mixes, and you can get a real plant out of it. That's one of the great things with working with plants, is that you can take cells from any tissue and you can regenerate a whole plant. That technology basically causes the DNA to be inserted anywhere in the plant genome. You don't really have a control over where it can go. And it doesn't allow you to make precise changes. One of the things that's happened in recent years is that there have been different technologies that have been developed that allow you to precisely determine where the edits to the DNA sequence will be. So some of these are things like zinc finger nucleotides, talons, and this new one, which is all the rage right now, called CRISPR-Cas. And this gives an idea of the types of things that can happen. Basically, what these things do is they make cuts in the DNA. And when the DNA is cut, if the cell is going to survive, it has to rejoin. It can rejoin either by a non-homologous end joining or by homologous recombination. And you can add other pieces of DNA. So this indicates, you determine the actual site where the cut will be made. If you provide DNA that is, if you don't provide DNA, then you can actually make a deletion of the region around that area. If you provide some donor DNA, you can have an insert of a gene. If you provide DNA that is homologous to these regions here, you can modify the sequence of the gene, or you can insert the gene similar as you did here. So this allows you to do a lot of really interesting types of modifications. This is a paper that just came out, high-frequency precise modification of the tomato genome. There are no genetically modified tomato on the market, but there's a lot in research labs because they're really good experimental tools. There's other ways that can do it, and there's companies that are based on this. This is Cebus is a company. Basically, what they're able to do is they're able to take pieces of DNA, single-stranded pieces of DNA, or sometimes, in some cases, people use pieces of RNA, and they target for a certain region, and they can make as little as a 1 nucleotide change. And they've been able to make different types of modifications. So the question is this any different than another type of breeding technique or a mutagenesis breeding technique? There are thousands of plants, crop plant varieties that you eat every day that have been produced through mutagenesis. So that is using chemicals or radiation in order to change randomly the sequence of the genome. I think that being able to precisely make the change is probably a better way to go. At least it should be more predictable. Okay, so what types of traits should we be interested in? Well, if we go back to how we started this conversation, one of the most important things is yield, right? We want to be able to get cultivars of crops that are optimized for different regions of the world that are able to do really, really well in those conditions, and preferably without adding too much fertilizer. We'd also like to reduce undesirable metabolites. So an example is cassava. Cassava is a really, really important crop in Africa, but it's got a high level of cyanide. And so raw cassava can't be fed to livestock, for example. It'll make them really sick. Microbial disease resistance. I'm doing some work in Ontario greenhouses. Canadian greenhouses are over a $1 billion industry. They lose 15%, it's estimated, of the value of product by having to deal with things like microbial diseases. And there's not really anything that you can do except try and prevent the microbes from coming in. Insect resistance, more efficient nutrient use, that's related to yield. We also want to improve nutrient use so that it doesn't go out into the environment and cause things like dead zones. Wouldn't it be great if we could have corn and rice that made their own nitrogen? Drought tolerance, salt tolerance, antifreeze. Drought tolerance and salt tolerance, this is pretty important for being able to use marginal lands. And as we have to deal with climate change, we're going to have to deal with more drought, like they are in California. This is also an important problem in many other parts of the world. Biofortification, can we actually get vitamins that are not present in certain food? Can we have it present so that you'll have a better nutritional profile? And then, of course, this really shouldn't be the last thing on the list, but improved flavor is something that's really important. And there's some people that are actually working on improving the flavor using a lot of molecular biology techniques, and then going back and using traditional breeding to make something that will be palatable to the public. Some of you might have heard of the Arctic apple. This is a non-browning apple that has been produced in Canada. There's the innate potato, which is also non-browning. It also doesn't make acrylamide during cooking. Some people think that acrylamide might be a carcinogen, but some people are afraid, like McDonald's, for example, won't use this because they're afraid that their customer base will not want to eat McDonald's French fries if they think that this is not a natural potato. And this is an example of what an infection can do to your crop. These potato plants that survived are the ones that have late blight resistance. All of the other ones have been decimated by that particular disease. The final thing that I'd like to do is just mention to you this synthetic biology, because this really has to do with opportunities for young people. So every year, there are 259 there are many teams from around the world. This was their 10th anniversary this year that converge in Boston for a competition. It's a jamboree. This past year, there were 259 teams with over 2,700 participants. And basically what they do is they work on a project of their own design for the school year leading up to this. So they work for almost a full year, and then they present it in a competition. And at Waterloo, we have a very successful team. Here they are presenting. And they actually worked on a system that could use genome engineering in order to reduce viral infection of plants. So they're doing cutting-edge research, but they're doing it in a way that is collaborative, and they're doing it in a way where they teach each other, in a way that's interdisciplinary. We have people from math, we have people from biology, we have people from arts, from communications, all coming together in order to work together as a team. And it's really one of the most fulfilling things that I have done as a faculty member, seeing these young people carry out this type of work for no credit. They do it for no academic credit. They do it because they want to learn how to be a scientist. They want to learn how science is done. This is my research team who does the research in my lab. And thank you for your attention.